Okay, I just put on the recording. I promise that we'll start in a minute, and we'll give a couple of minutes time if someone's joining us, but I think we can regardless make a start at this point. I really appreciate you joining me here tonight. I know that everyone has full packed schedules. So what I'm planning to do today was we'll take a little bit of a time to go through the material from the review of exam 2 and lab practical 2. So those grades have been uploaded and they're up there in terms of corrected and final ones. But I want to go through some of the key themes that came through from those things that were common, either things that students missed or common things that we might want to check in other ways to make sure that they are clear for everyone. So I want to spend a little bit of time on that, and because of that, I want to keep a little bit of a tight rein on the time. So that's why I'm making a start immediately. I also want to go through today, especially the material from the fluid, electrolyte, and pH balance chapter, which is the chapter that we're covering this week. That chapter on its own, it's really not that long, or there's not that much of a new material. A lot of that will be familiar to you from your BIO 181 or BIO 156. But because I want to have the time to cover that, I'm just making a start at this point. And just to welcome anyone who joined us, you did not miss anything. We were just getting started. And all I really said was that today I want to spend a little bit of time going through the previous exam and previous practical, and then go through the material from this week's chapter. So that's as far as we got. If you do have any questions at any stage, feel free to shout out. But if I don't hear anything, it doesn't mean that I'm not welcoming the questions and I'm rushing along. I just want to make sure that I don't take too long of your time today, so you get to your evening and onwards with those. So that's the only reason why I'm making a start. But at any stage, feel free to just let me know through the chat or through turning on your mic, and we'll make a start, or we'll tackle whatever comes up. We'll make a start with the exam 2 and practical 2. I was gonna apologize. I was gonna apologize for if you hear from the background noises of the birds. I have actually a bunch of new chicks in my room. I keep chicken, but I was able to weasel my way out of there with the other room, so it should be fine. But if you hear little sounds on the background, that's just them. In terms of the practical 2, I just wanted to highlight a couple of important generic points. Do remember that, and I think that both of you did really well on this, so this is more of addressed to anyone who's watching the recording, but I regardless want to cover everything. Do remember that I do give points for just trying in the practical. So if you don't know the particular answer, or if you don't know the particular structure that you're looking for, you can tell me structures around it or something about the physiology or something. So I give points whenever I can. It might not be the full points, it might be partial points, but as long as you give me something that I can give points out of, I will definitely try to find a way of doing that. So I always emphasize to try to get at least those partial points. In terms of the chapters that we covered, the most generic things that came up in terms of the heart chapter and content on the heart was that we want to be certain about the chambers that make up the heart. And there's many different ways of drawing the heart. What we're going to do here, I'm just going to draw the most simple version, which I showed you how you can draw a heart. And that's simply a box that's divided into 4. And we have right side and we have left side. And remember that the right side is going to be the patient's right side and left side is going to be the patient's left side. And then we had atria at the top, so more superior chambers are atria, and more inferior chambers, the ones at the bottom, are ventricles. And typically what we find is that the left ventricle especially has a thick wall. So that's the main chambers that we want to know from the heart, that are kind of a requirement to know where they are located, so we can build rest of the material on that concept. In addition, one thing that became obvious, and again, I'm not saying that necessarily with you two, but in general in the class, and that's important for us to be clear of, is that we want to be sure where some of the major vessels come from, where they go to in the heart. So we have 2 vessels coming from the body to the heart from the systemic circulation. So from the above comes the superior vena cava, and from bottom comes inferior vena cava. And those bring the blood back to the heart that has been around the systemic circulation. And as a result of that, that blood is often deoxygenated, so low in oxygen. A couple of other major vessels that I want to highlight, leaving from the right ventricle, we have our pulmonary trunk. And that, of course, goes to the lungs. So that's from the right ventricle. And then leaving from the left ventricle, we've got aorta. And that goes then to the systemic circulation. And on left side, we're of course then going to have, as a result of that, oxygenated blood, since it's coming back from that blood is coming from the lungs, so there's a high amount of oxygen there. A couple of other things that were things of word of noting, coronary arteries, we discussed about those, and we discussed that we've got 3 that we need to know at this level or at this stage. I'm not asking for anything more than that. So we have our right coronary artery, which if we have here the aorta, here we have the heart, and here we would have the aortic valve. So the right coronary artery comes out on the right side of the heart, right after the aortic valve. And then left coronary artery comes on the left side of the heart. Left coronary artery divides then almost immediately to anterior interventricular artery, which is sometimes also known as LAD. I think LAD is the more common term in cardiac sciences and cardiac medicine than just a traditional anterior interventricular artery, but both are good to know. And then we have the left circumflex artery, which will then wrap around the heart's anterior side and go to the posterior side and then supply the posterior aspect of the myocardium. So those were the coronary arteries. In terms of the histology, I don't have there any really specific examples. Just a reminder that try to be as specific as you can whenever we talk about histology. It is just as important as those gross anatomy structures. I personally find that things that I can see with my naked eyes, touch with hands, come easier for me. So I work a lot with the gross anatomy structures, but it doesn't mean that the histology wouldn't be an important part of physiology and anatomy. So we do need to cover those as well. Well, let's have a look at a couple of examples about the practical things that were common, things that we should definitely note. So sometimes with the valves, especially, we can have multiple names, and it doesn't just have to be the valves. There can be other structures. But when we have multiple names, all I'm asking you to know is just one of those in our practical exam. If you know any more than that one, that gives you a chance to earn extra credit. And the extra credit really, I never deduct points if you give a guess and say something and it's not quite right, as long as you have one correct answer there. So for example, here, let's just have a look of the heart that we have first, the model. So we would have our right atrium and right ventricle here. On the left side, left atria and left ventricle. And we see that there is this valve that's being highlighted, and we're being asked that what valve is that? Well, the first good clue here is the blue color of the vessel that comes out. So that's going to be our pulmonary trunk. And pulmonary trunk is right ahead of the pulmonary valve. So if you said that it's a pulmonary valve, that was full points. You could also say that it was a semilunar valve. That would be full points. If you said both, then you got an extra point for that, in addition to the maximum points. So there's always a chance to kind of play it smart if you know more than just one answer. Other thing that, or other place where I saw a good example of that was again with another valve. Here, let's again map out what we see. We see our right atrium, right ventricle, left atria, and left ventricle. And we're now looking at this valve that connects the left atria to left ventricle. And one way to answer that would have been that it is the left AV valve, so left atrioventricular valve. The other way you could have answered to that would have been that it's a bicuspid valve, telling how many cusps we have there. We have 2 cusps. Or the third one would be that it's a mitral valve, which is another term that's also often used. So 3 options there. And as long as you gave me one, you got the full points. And if you gave me more than that, you could earn extra credit based on how many of those answers you gave. Sometimes we also have in anatomy and physiology, and especially on these practicals, very specific structures, detailed structures that we might be looking for you to highlight. So, of course, I realize that it might be a little confusing, but just get into the habit of trying to look that is there something detailed that we're pointing, or are we pointing to the general entire structure? So what we see here is our right atria, but within the right atria, we have circled a particular part there. It looks a little bit like a depression on the inner wall of the right atrium. That is actually gonna be our fossa ovalis. And if you don't know what a fossa ovalis is, I'm just giving a really quick explanation. So we remember that we had our heart, which we could draw as a box. And there we now see that within the right atria, we got that little depression, that indentation that's somehow important for us to know. Well, before you are born, so for the duration of time before you come to this world physically are born, there is actually an opening between the right and left atria. And this allows the blood to travel immediately, directly from the right atria to the left atria. And if you think what structures we have between there, that would be, of course, the lungs. So from the right atria, blood normally goes to the right ventricle, and from there to the pulmonary trunk. Well, if you're not born yet, you're still within the mother's womb, there's not going to be lungs that would be functional. There's no air to breathe. So this method allows us to bypass the entire set of lungs. And there's a couple of other adaptations that contribute to this as well, but it allows us to bypass the non-functional lungs before you are born. So the moment you are born, that's when the lungs start working. And at that moment, the structure that we've had a look now, the structure called fossa ovalis, that closes. But we can still see, even in an adult heart, on the right atria, a little indentation on that wall. And that's the opening, what used to be fossa ovalis. And sometimes fossa ovalis doesn't close, and that might be a problem. And then we might need to have a look what we could do at that point. A couple of other things that I wanted to share with you. This is more of a reminder. I'm sure you remember that spelling, I never deduct any points if you misspell something in the practicals. So really, it's not an issue as long as you give me something that I can feel with good conscience that, oh, I'm sure that they knew what it is, even if the spelling is not completely correct. I'm quite happy to accept that. Here, what we see is actually let's start by looking again what we see, the basic structures. We see our left atria and our left ventricle. And some of the students told me that, yes, this is the left ventricle that has been highlighted here. And in a way, you're right, but I think that the left ventricle, if we were asking that, we would have highlighted even larger area of this view. Left ventricle is the entire area. Instead, what we see is that we have highlighted only a particular part, and that part is the inner wall of the left ventricle. And that inner wall is trabeculae carneae. And what that inner wall does, there's this Bambi-like structure of the muscle, and that structure of the muscle, that pattern on the surface of the interventricular wall, prevents blood from staying still there. It ensures that all of the blood gets squeezed out when the ventricle empties, because if we have blood sitting still for a long time somewhere, it will start clotting. And we don't want that to happen. So that's what the trabeculae carneae serves us for. It helps us to ensure that the entire ventricle is emptied. I got some of the students who proposed that maybe it would be papillary muscle. You remember that papillary muscle was the muscle that connected to these chordae tendinae that came out from the valve of the heart, or heart valve, and after that we have a chordae tendinae which connects these, there's chordae tendinae, we have the papillary muscle that connects to the ventricular wall. So that was what some students offered. But if it was just the papillary muscle, it wouldn't be this large area that's highlighted here. So again, depending on a little bit of what kind of the answer was, it did not yield full marks. It could yield partial marks in some cases. Another thing, again, here, I'm just simply showing any spelling. I really don't mind as long as it makes sense to you and me, as long as I can say that this student knows what they're talking about. How you spell it, I'm quite happy to go with whatever version you end up with. Sometimes what we end up finding in these practicals is as well that we have a structure that we ask. Like here, for example, we are showing the pulmonary artery. So, of course, let me just get the correct pen. Of course, here we see the pulmonary artery. If you answered pulmonary artery, you got full marks. However, some students were very observant and noticed that we were highlighting or pointing to this particular part of the pulmonary artery, and that would be the left part of the pulmonary artery. So that was again a chance to earn some extra credit if you included more information than what was required as the minimum for full marks. Let's have a look at what else I have here. This is a key point, and this is not really particular for this practical. It comes up in many instances outside just this practical. But we want to be certain that which one is radial side and which one is the ulnar side from the distal part of the upper limb. So sometimes students learn these by learning the bones and then just are able to use that knowledge as they move ahead in their studies. Sometimes students learn these when they learn the nerves, radial and ulnar nerve, and are able to use that information moving ahead. Sometimes students learn these when they learn the blood vessels and are able to use that knowledge moving ahead. But at some point, you need to be making sure that you learn the difference between radius and ulna. So what we find is that the radius is on the thumb side of the individual who is in an anatomical position. So it would be here. And the ulna then is on the pinky side of an individual who is on the anatomical position, or of the upper limb that's on the anatomical position. I asked my students back in the day that, well, what do you do to remember it? Because I have my own ways, but mainly for me, it's memorization. I've been doing this for a while, so I just remember them. But one student gave me a very good mnemonic that I think that I wanted to share with you guys. So if you're struggling to remember which one is radius, which one is ulna, you remember that when you put your thumb up, you're like right on, and you're right on for radius. So on the thumb side, you will see the radius, and on the pinky side, you will see the ulna. So I don't know if that helps you. I thought it was a fairly creative way to remember these, but just make sure that you learn those because they come up so often. It's worth learning them earlier than later. What else could be worth pointing out from the practical? Try to give as complete answers as you can. So I know that a lot of the time when we are covering material in a class or when we are going through material in other instances, we just end up kind of abbreviating things. So here, for example, we see a blood vessel, actually an artery, running on the pinky side. And as we just learned, that's going to be the ulnar artery. But often I might, for example, say, or you might see in your own notes that it's just abbreviated. Oh, it's the ulnar vessel. Make sure, though, when you're taking an exam that you include the artery. So that way you get the full points. Sometimes if I had only ulna written, I would have given full points, but then if you gave me full answer, that it was the ulnar artery, the first time you might have gotten an extra credit for 1 point. Second time that happened, you could have gotten half a point. And from then onwards, no extra points. That's simply because I wanted to go soft at the beginning of the course. I didn't want to be too harsh. I think in university, you just wouldn't get the points because it's not the full answer. So always try to be as complete with your answers when you're taking an exam as you wish. If you prefer, like in my own notes, I often do ulnar A, just to remind that it's an artery. And then it would be V if it was a vein. So that might work as well. I would totally accept that for the full points. This one, I think, is a pretty good point to take on board, both on this practical, but in any practical. Sometimes structures that we're looking at are named based on the other structures that we might have on the same area. So it again goes to that, learn the material well once, and you'll then just have smooth sailing through the rest of your time studying anatomy and physiology. So for example, we have learned that we have clavicle, which is the collarbone running here. So if we have a structure that's described as a subclavian, that tells us that it's the under this clavicle. So rather than just learning a completely new term, subclavian, because you already know the clavicle, it's easier to remember, well, let's just think of what subclavian means. It means under the clavicle. So that's the vessel, the vein that we see here, for example. Sometimes the location plays particular role, that you might have a location where blood vessels are especially tricky like this. The vessel is called one thing at one point, but as soon as we move a little onwards from that point, the name of that vessel changes. So here we see the section of this vessel that we looked at earlier on, just in the previous example, that was subclavian. But as soon as we move a little bit closer to the heart, this name of the vessel changes to brachiocephalic vein. So it really matters that what part of that vessel we look at, even though the vessel might be continuous in terms of its appearance. We name often parts of vessels differently based on their location. Another example that I have here goes with the common carotid artery. So common carotid artery is the section that we see at the level of the neck. And when it divides at the level of the neck to external and internal carotid artery, those are the separate names from there. But before that, when they're just joined, it's going to be common carotid artery. And the reason why I highlight that is this next one, that here on this question 19, I got many students telling me that this would be common carotid artery, where it's not yet on the level of the neck. What we see here instead is our brachiocephalic trunk. So the first, yes, you have taken the practical exam too. So we're simply going through reminders of what we're when I've graded the common things that needed to come up. So I'm just giving a quick feedback of that. So I think that on this model, it's challenging to see the brachiocephalic, but if you look at the proper dissection, you will actually see it more clearly. So brachiocephalic is the first branch from the aortic arch that comes off from there. And because we're just going through a review, I don't want to spend too much time on this, so that's why I'm just going through them quickly. Sometimes we have same term that we use for many different systems, that it doesn't even need to be the same system, but within a certain region, we keep seeing that the same term comes up. So what we see here, we remember that the term for armpit was axilla. So we have axillary artery, but we would also have axillary lymph nodes here. So as long as you learn those regional terms, it makes the naming of structures so much easier. So I'm just trying to really encourage you to get the basics right, and it's going to make your life easy. Sometimes the location, if you give me further detail, I can give you more points for that. So here, for example, we saw the tonsil, and this particular tonsil was pharyngeal tonsil. So if you just said that it was tonsil, you got the points, but if you gave me more detail and told me that it was pharyngeal tonsil, you got, I recall, extra credit. That's because we've got so many tonsils, I wouldn't know, do you really know this structure? So if there's something that we have multiple pieces, then try to include a little bit more detail of that. And sometimes this goes to what I said earlier on about having extra detail, which gains extra points. Sometimes we have structures such as here we see thymus, which is very prominent in a young individual, but on an older individual, it becomes harder to spot. But if you did spot the thymus, you got the full points and you're all set. But if you spotted it and told me that the reason why it's hard to spot is because it's these remnants of what used to be more easily visible thymus, then you got extra credit for that as well. One more thing that I want to make sure that we are certain, because this came up on few students, the difference between pulmonary and aortic valve. And again, I'm just gonna jump and draw the very, very simple diagram that we drew before. Here we have a heart, we have our right side, and we have our left side, and we have our atria, and we have our ventricle on both. And remember that from the left ventricle, we will have our pulmonary trunk coming out. And from the right ventricle, we have our pulmonary trunk coming out, apologies. And from the left ventricle, we've got our aorta coming out. So for that reason, if you end up seeing a valve here, if it's on the right side, it's gonna be the pulmonary valve. And if it's on the left side, it's gonna be the aortic valve. So just make sure that you're clear of those, because those sometimes get students confused. And I think that this is a much nicer diagram of what I just tried to explain. So in addition to the pulmonary valve from the right ventricle, I've added that separating the right and atria and ventricle, we've got the right AV valve or tricuspid valve. And then in addition, separating the left atria and left ventricle, we've got our left AV valve, sometimes known as bicuspid and sometimes as mitral valve. And of course, we've got the aortic valve leaving the left ventricle. So that was the practical. And I still want to say, I know that we have a full chapter to look through, but I want to say a couple of words about the exam. So in terms of the exam, what was kind of most common themes that I want to remind you, if there is an extra credit question, I do encourage you to tackle that if you have time, because I will never deduct points. Only thing you can do is gain more points. And even if you don't know an answer to that exact question, tell me about something else and I'll give you some points out of that. In the exam, we had pretty much 3 topics that we covered for the exam too. We talked about the heart, blood vessels, and lymphatic system. So for the heart, the big thing that I wanted to make sure is that we remember what kind of blood we have running on each side of the heart. And again, if we think back when we first started, we might remember that I used a poetic expression that the heart is like 2 hearts, human heart, beating as one. So we have 2 sides to that. So that is important when we think of what kind of blood is flowing there. And if we look at the right side, we have deoxygenated blood coming from the body. So this is where we have very little oxygen there. And that travels through the superior vena cava and inferior vena cava to the right atrium, from right atrium to right ventricle, from right ventricle up the pulmonary trunk. And all of this is gonna be this deoxygenated blood. On the left side instead, we will have reoxygenated blood, or sometimes just it's called oxygenated blood, where the oxygen concentration is high because blood is now coming from the lungs to the left atrium. It flows from left atrium to left ventricle and then out from the left ventricle to the aorta. Ooh, I don't know where that line, sometimes my screen gets a little bit sensitive, so it draws lines of its own. So what I wanted to say there is that you notice how on the right-hand side, it's going to be all about the deoxygenated blood, whereas on the left side, it's going to be reoxygenated. And this color difference was what we often use to guide there. The other question, and I think I mentioned that I might ask this in an exam, was that what was the first vessel after aortic valve? And often students go and work really, really hard learning blood vessels, and they want to show off that they've learned. So if we look at the aortic arch here, we have 3 branches coming off from there. So often students are really good about it and tell me that this first branch of aortic arch is then, as we saw here, I'm trying to find I probably have it coming up later on, that it would be our brachiocephalic trunk. But that's not actually the very first branch that comes out of the aorta. So right here after the aortic valve, we've got our coronary arteries, our right coronary artery and our left coronary artery coming out. So those are the first vessels. So I was looking at that. That might be a little harder question, and sometimes students kind of get lost with that. When we are talking about some sort of an order of events, do try to memorize it as a pattern. For example, I asked about the electrical conduction of the heart, so you needed to know what part comes after what. And I've got a good example of that coming up. And I also encourage you to remember that there is this connection always between anatomy and physiology. So the form often defines the function, and then function might define the form. So if you're trying to figure out the function, think of what that structure looks like, what's characteristic to that structure, and that will help you to work that out. Let's move on to have a look at what we had main points from the blood vessel chapter. I wanted to make sure that you know the difference between arteries and veins. And the big difference there to remember is that arteries were thick, they were elastic, and often they're kind of a deep, protected within the body. So if there's a cut, that cut doesn't get to an artery easily, because if an artery gets cut, you usually are likely to just bleed death really fast. Whereas we saw with the veins that they were thin in terms of their walls, and we had the adaptation of valves there. And often they are very superficial. So that's the difference between arteries and veins. Sometimes, again, we have multiple names for the same structure, so any of those could be asked. And I really encourage you to use the mnemonics when I've given you some. Those usually help, or you can make your own. In terms of the lymphatic system, just 2 basic things that I wanted to highlight. I told that I'm going to ask, when does the interstitial fluid that we have become lymph, because their composition is the exact same. The only difference between interstitial fluid and lymph was that when the interstitial fluid entered the lymphatic system, it became lymph. And then I asked a couple of questions about the functions of the lymphatic system. So some students just weren't totally clear with that. Remember that, of course, the pathogens and protecting against pathogen is an important and most obvious function, but lymphatic system also plays role in distributing the fluids throughout the body, and also transporting the dietary fats, where the dietary fats were too large-sized molecules to get into the cardiovascular system, to the vasculature of the capillaries. So then the lymphatic system picks them up and transports them. So some examples, questions that are tough in the exams generally are when there's a question, what is not correct? So usually there, the easiest way to tackle these is by doing the process of elimination. So you look at which ones you can point that, yes, I agree with that, I agree with that, I agree with that. So here the big difference was that I had put in a mistake in the description of the apex. Apex, of course, points anteriorly, so to the front. It points inferiorly to the down, and it points to the left. So for that reason, this question or this statement had to be false. Sometimes it's not quite as clear that I'm highlighting the word not. Sometimes it might be termed like incorrect. So what answer is incorrect of these? Here I claimed that the young children, we can often put mechanical valves on them. And of course, we know that that's not true because mechanical valves don't grow as the young individual's size grows. For example, they're hard, so we would have a problem. We would have to do another surgery, which of course wouldn't be ideal. All other statements on this example were correct. Sometimes you get questions where I'm asking you to select all that apply. So if you have a question like that, more often than not, there are more than just one correct answer to that. So here, for example, I was asking which of the arteries, coronary arteries, supplies the anterior portion of the heart. And of course, the one that you have probably seen more commonly is the anterior interventricular artery. But the other name for that very same artery was the LAD, left anterior descending. So if you selected both, you got full points, the full 2 points there. If you only selected one, you did get partial credit, but just be on that lookout for that there might be sometimes a case that I'm asking you to share a little more than what's immediately obvious. Here, an example, I was asking naming the vessels that generate the second stop sound. So basically the vessels that close secondly in the cardiac cycle, and we will probably all jump into that. It's going to be pulmonary and aortic valves. That's correct. But the name for both of these, that covers both of these, is semilunar valves. So I was actually looking in this one, all three of them for full marks. Otherwise, you ended up with partial credits. This one's the question that I already discussed with you guys, that what's the first vessel after aortic valve, and that was of course our coronary arteries. So coronary arteries are immediately after this. Oh, I'm sorry, it's a tough question. So the coronary arteries are immediately after the aortic valve, and then we get the brachiocephalic here on the aortic arch. It's probably the toughest question, so don't beat yourself up if you miss that. That's you're still probably doing pretty good. I mentioned earlier on at the very beginning that sometimes I ask about the order of the events. So really, I personally just memorize them. But I would encourage, if any of you are visual learners, I try to also include some sort of a visual animation or something that shows these things happening. I remember that for the electrical conduction, I had that showing how it proceeds through the heart. So if that helps you to remember, go for it. It can get a little bit muddly in the head when there's a lot of terms, but just try to remember. So here, for example, the big thing was just to be certain that you remember the right order of these terms. Sometimes the questions might be questions that you will benefit from thinking of the old material that you have covered before. So we have talked about the action potential in the nervous system at the very end of the chapter or at the end of the BIO201. And there you remember that we had the resting potential about -80 millivolts. And then we had the depolarization, where we approached the 0 millivolts and even shot over that. Then we had a period of plateau. Then we had repolarization and undershoot. which was the refractory period, and then we got back to the resting potential. So if you knew that, that would make it easier to spot that even though, yes, there is a slight angle in this diagram that we see, but it is close enough that we can assume that it is going to be this plateau stage. So another tough one, so don't feel too bad if that was something that threw you off. Sometimes we have multiple names for the same structure, like we saw earlier on here with the pulmonary and aortic valve, also being known as semilunar valves. So there's nothing too much of exciting that I can share about that, but just try to learn all of the names the best you can. In terms of the exams, I totally can ask diagrams. So some students find that they're more visual, and this would be working out great for you here. So here I had different types of capillaries that I showed. The very first one that we see here as option C is going to be the fenestrated capillary. You remember the word fenestration came from the French word for the window. So there's small windows on the wall of these capillary types. The next one, the letter B on this diagram, is continuous. So there's no gaps at all. And the A with the biggest gaps is going to be sinusoidal. So if you think of what sinus means, it's a big opening somewhere. So it's bigger than a window. So on this one, I had kind of mixed the order up a little bit so that it wouldn't be too easy to name those. But if you knew those, that wasn't too tough of a question. Sometimes questions are follow-up questions from what we saw earlier. So in the earlier one, if you did notice that continuous capillaries had these big parts of the wall missing, then you could work out simply by the laws of physics, if there's a lot of wall missing, there's a lot of then friction. It's not going to be smooth flow, and that's why the blood is going to flow really slowly on that area, and that allows a lot of exchange to happen at that site. So the major site of exchange was going to be the sinusoidal capillaries, these capillaries with the biggest gaps on the wall. Mnemonics was something that I encourage you to use. Sometimes there's just a lot of information and it becomes a little hard to remember everything. So for the signs and symptoms of the shock, I use the mnemonic when I teach you of TC fats. And since I come from UK, we always like to make fun of our police constables. So that's where this abbreviation comes. So the signs and symptoms of the shock is pale, cold and clammy, fast and weak pulse, or fast and superficial breathing. Patients who are going into shock are often agitated and they might be thirsty. That's quite common as well. And that's all indication that they're about to go to a shock. So again, I think that when you get a big list that you have to choose which ones are correct, that becomes a very intimidating question quickly. But if you remember a mnemonic from that topic, then it becomes easy because you just tick off the ones that match that mnemonic. Sometimes there are simple definition questions, such as when the interstitial fluid becomes lymph. Well, it was when it entered the lymphatic system. So that's what I was looking there. And that really concludes our review of the practical 2 and exam 2. And I know that we spent a little bit of time on that and I'm kind of cringing. I don't want to keep you too long. So I'm going to try to keep going. If you have any questions of those, I absolutely am happy to answer. I just propose that maybe we do it at the very end because I want you to be able to get to your rest of the evening. So jumping on to what we are doing next week, and that week number there is wrong. That will be, of course, the week 14. So the exam 3 and practical 3 are going to be available for you for the all week. So you can complete them at any stage of the week. I don't have new assignments for that week. All I want you to do is to tackle the exam and practical. I offer for the exam 3 2 days of review sessions. So 2 review sessions on Monday and 2 review sessions on Wednesday next week. So once at the morning at 10 o'clock and once in the evening at 8 o'clock. And what we do for those, we use the study guide, which is available for you, but you remember that there's gaps. And the only time that I really give the answers to those gaps is during these review sessions, or if you choose to watch a recording of these review sessions. And remember that we're covering the chapters 22, 23, and 24. So the respiratory system, urinary system, and water, electrolyte, and pH balance. So what we have covered after the spring break. But the material that I'm asking you to know is only the lecture video content. You don't need to know anything outside that for the exam. The exam is going to be 50 multiple choice questions, about a minute per question. I think it's 60 minutes in total, unless you have an accommodation in place. It is open book. So for God's sakes, if you feel that having study guide that's perfectly complete, it helps you, use it. So the study guide contains every single question in the exam. I might adjust them a little bit, mix them up a little bit, but every single question is pulled from the study guide. In terms of the practical 3, it's going to be the same. So next week, available for all week, entire week. You do it whenever you're ready. There's a couple of review guides or lists of structures that you need to know, but I really encourage you to use those practice versions that we have available. I had a look through it. There's 50 questions. They're all these filling the blank. Again, spelling doesn't matter. They're going to be different organs and very specific structures. And you have 60 minutes unless you have an accommodation plan. And again, it's an open book test. So if you take good notes, some students take screenshots, some students take pictures of the practice versions, whatever works for you, make it easy for yourself. So I'm just encouraging to optimize your chances of success. But that's about next week. About this week, let's jump to that and have a look. So this week, I don't have a huge amount of stuff for you to do. Everything that I'm asking you to do is due at the end of the week. And again, you remember that I'm very flexible with extensions. I have a couple of emails asking for those, but I just was in a commitment to get the exam and practical graded. So now I can jump onto those messages that I have. But what I'm asking you to do is to learn, smart, and quiz. And those are what you have done before through the publisher, nothing new there, through the connect. The new part for this week, an activity that we have never done before, is this critical thinking question. And this critical thinking question is something that you will complete through Canvas. You might wonder, what is the critical thinking question if you haven't seen these in other courses? If you have, you are already clear, but I'm going to cover it very, very quickly. So in every course in our college, we will have some sort of an assignment that tests critical thinking. So it's making students think a little bit beyond just the basics. So in this week's critical thinking activity, I'm asking you to complete 2 case studies or scenarios, if you wish. So you get to be the doctor or a nurse or whatever healthcare professional you want and work out what's happening with the patient. So remember that there's 2 of those, and I'm asking you to do both of those. There's 1 on each page, and I think their numbering is weird, that there's first question 2 and then question 1, but as long as you get both of them done, you're fine. How do you find these critical thinking questions? It's all in the Canvas. There is going to be a link. That's a hyperlink or a link that opens up a file that says critical thinking assessment. You complete that, and then you upload it back to me. If you prefer, you can just type in your answers to the textbook on Canvas, but remember, Canvas sometimes logs out, so I would at least type them in at Word, but some students, or most students, find it easier to fill in the answers to the actual Word document and then just upload it for me. But I will grade them from Canvas, so you will not find the critical thinking question from the Connect. Make sure that you take it at the Canvas. One more thing that I want to mention quickly about that is that there is grading matrix that's actually available for you. So if I was a student, I would check out that grading matrix when I'm doing the questions or before that what is the professor looking? What do I need to know so that I get full points? But this is the page where you land when you click on this week's critical thinking question. And as you'll notice, it tells you a little bit about that. And then you see this link here. And by clicking that link, it opens the Word document where you have those two scenarios. And you have a choice to either fill in a Word document and you save it again and send it to me and upload it to there on the Connect, or you can just type in your answers, whatever floats your boat. This is a part of the grading matrix that I've included. I didn't get a chance to upload all of it on this presentation, but it's all there on the Canvas for you to have a look. And it shows you question by question, what do I ask from you for you to get full marks? So for example, on this question, you need to get 8 or 9 of the likely or unlikely questions correct. So you choose, is it true or false, likely or unlikely, the statements. And if you get 8 or 9 of them correct, you get full points. So you don't even need to get all of them correct. If you get 7 or 6, you get 3 points instead of 4. 4 to 5, you get 2 points, and then you get 1 point for any less than that. Another example here again for full points, you need to identify 8 to 9 problems that the patient might have. And then the number of points goes down if you identify any less. So that might help you. But what I'm going to jump into now, because I am aware of the time, and if you do need to go, I will not take that personally. I totally understand. But I will record this and it will be made available. And I'm trying to go through the fluid, electrolytes, and pH chapter a little bit quicker. So we're going to have a look of some of the key points of this week's chapter. And like I said, it's not in that much of a detail. So I think that we can pull it off without taking too long. So let me just jump out from this present. And I'll open the other presentation so we have a chance to go through the chapter material for this week. And you remember that there is always the full lecture material as well, available as those recorded videos, but I try on these review sessions to give a brief kind of a walkthrough of the very, very, very key points that you need to know. And let's get this other presentation that I just got open for us shared. So you should be able to see, I spoke to some of my students. They asked about the exam, how to succeed in an exam and things like that. So I just say that make sure that you're well rested, well eaten before the test. Since these are open book tests, make sure you have your notes around you and you're somewhere where you won't be interrupted. These are one-sit exams a lot of the time when they're open books, so you can't really leave and come back. During the test, try to answer all of the questions. I never deduct points if you answer wrong. And give me at least something. And then when you have had the test, we just did what you should do, review where you went right and where you went wrong. I take a lot of time to write you feedback as well on your tests, so check that out. But we are jumping on to the chapter material for this week. So we're going to talk a little bit about body fluids, where they are located, what are the key properties of water, how water balance is maintained, and then we'll see the connection between electrolytes and tonicity. And finally, we'll finish off looking a little bit of the pH. A lot of this will be for you review from the material that you have seen in BIO201 or in your other previous courses. So just use use your own judgment that how much detail you have to go into to really grasp this chapter. There's not too much of a new material. I wanted to start just by showing a little bit when you're wondering, why do I need to know about the fluids and electrolytes and acids and bases and so on? I want to be a nurse or I want to be a medical doctor. Well, really, a lot of the time we do labs where you get values of these. So you need to know what's normal so you can tell what's abnormal. And a lot of the time on those lab reports, you will get reference ranges, which would be typical. And then you get the patient's value. And then you start working out if something's off the range, what would it tell us? And that's very much what you will see on this week's critical thinking assignment on those case studies or scenarios that you'll work through. So this is a big part of working in the medical field. So even though it feels like going to the basics, knowing the basics is needed before you can know the advanced. We'll also have a look of a little bit of a couple of clinical conditions at the very end. So this all is really important so you can understand the patient's basic lab test results. When we talk about the water and why water is so important, on BIO181, BIO156, I normally start talking with the students just about the planet in general that we live on, the Earth. And majority of the Earth's surface is covered by the water, by seas. So it is something that's vital for us to have life on the planet. But it's also the fact that we find that most living organisms are made mainly of water. On humans, it depends a little bit on the textbook that you'll be using. You'll see numbers between 60 to 75% of the human body being made of water. There's actually some patterns that we see. We tend to see that males have slightly more water in their body than females, and that's because the female body tends to be a little bit higher in the fat content. They have less skeletal muscle, so that explains that variation. What we see, what I think is exciting or interesting, is that infants, newborns, have the highest amount of water in their body. And that's because, again, they have low body fat percentage, they have low bone mass. And then if we go to the very old people, this is a bad joke, but people literally dry up when they get older. So the amount of the water in your body becomes lesser. So I have done here, I think it's just memory-wise, a little bit of comparison between grapes and raisins. So let's have a look where we find fluids in the body. Well, about 2/3 of the fluids in the body are located inside the cells. So we call those fluids intracellular fluids. And then about 1/3 of all the fluids is located outside the cells. So either on the space between the cells or somewhere else that's not inside the cell. That's extracellular fluid. What I wanted to say here, I'm just going to take a very, very brief time and you can relax. This is absolutely not in your exam, but I wanted to give an example why it really matters where the fluids are. Are they inside the cells or are they outside the cells? And the example that I was going to share with you is actually related to my own work and what I do and did mainly before moving to the teaching field on research fields. So my background is doing medical imaging and especially MRI imaging. And what we see here is an MRI of a patient. We see the chest bone, we see the heart, short axis view of it. We see the left ventricle, thick muscle wall there, and thinner muscle wall of the right ventricle. We see liver underneath it. We see the lungs around and above the heart. So what I do and why really we need to know where the fluids are, I'm interested of patients who have heart attack and part of their heart muscle wall is dying. So what we use typically in traditional MRI imaging of the heart, we give the patient a contrast agent, gadolinium, and it shows up as bright area on these imaging sequences that we use on the wall of the heart if that part of the heart is dead. So this individual has had a blockage in one of the coronary arteries, and as a result, this part of the heart muscle wall is dead. Well, the good news is that you actually can have like about half of your heart muscle wall dead and still be able to live. You might have to limit your activities, but you can still live. And just as a side note, I think I said that before, but this is not in the exam, so you don't need to worry. I just want to show why where the water is matters. Well, that's what we did traditionally for the longest of time. And doctors then tried to figure out, oh, do we think that that area is growing, or is it the same size? Should we do an invasive surgery or not? Well, what my work has mainly focused is these novel ways of taking MRIs, producing novel maps. They're a little bit more colorful, fancy-pantsy, but that's not really why we do them. What we see on these is that the signal intensity varies. This is the same patient, same heart, exact same location, varies, and we see this brighter yellow lighter part, which is significantly larger than what we saw here for the dead part of the heart tissue. It's larger on these two types of maps that we can produce and that I'm especially interested. The fact that it's larger, it tells that this area is dead or will become dead if it doesn't get blood. And how we know that it will become dead is that any tissue that is at the risk of dying in the cardiac muscle wall, actually water escapes the cells and goes outside the cells to this extracellular space. So what I'm taking pictures here, rather than of dead muscle, is taking pictures where do we find a lot of water outside the heart muscle. So we're seeing that there's more area that will die eventually if we don't do some sort of an invasive intervention. And this is what's very useful for doctors. So what we want to do is we want to calculate that the risks of doing a surgery and the benefits of potentially salvaging the part that would otherwise die. So that's what my work has focused on and why it really matters where the fluids are. The fluids in this case will tell whether the patient's heart muscle will live or die. So that's what we know as extracellular fluid. Extracellular fluid amount goes out of or goes up, water escapes the cells if that tissue is at the risk of dying in a heart. But like I said, that's not in an exam. I just wanted to show you why these basic concepts matter. They are the concepts that science is made on. Okay, now we return back to the material that is part of the exam material. So let's have a look at some of the key characteristics of water. And a lot of these we have already covered in your previous courses. So a lot of that is not going to be terribly new. So I'm going to go through it rather quickly. Well, first characteristic of the water is that it works as an excellent transport media. So in humans and other animals, blood is mainly made of water, and it's the blood that allows us to transport nutrients, waste products, gases, hormones, many different things. So water is really good at transporting stuff. Well, water is good at transporting stuff also in plants. They might not have blood, but it is the water that picks up all the nutrients from the soil and transports them within the tree structure until to the areas where we do the photosynthesis. So again, water plays a key role on transporting materials, not just on animals, but also on plants. The next two ones that I want to talk about are cohesion and adhesion. And I'm talking about them together because there's a lot of overlap. So cohesion refers to the fact that water molecules tend to stick together with other water molecules. And this is what enables us, for example, if you take a glass and fill it up so that it's overfilled and you see that the water level comes a little bit above the rims of the glass. It's kind of a little bit of looping on there. Well, it is this cohesion between those water molecules that prevent them from sliding all down from the glass. So it's able to form this little dome and keep greater amount of water in that glass than what it should. Adhesion is another type of attraction of the water molecules, attraction of the water molecules to other polar surfaces. So for example, here we see the spider's web and how water droplets have attached to that very tightly. Well, that's because the spider's web is very polar, so water has been attracted to that. The next one that I wanted to talk a little bit about is the surface tension of the water. So because there is this tendency of cohesion taking place, some animals, especially bugs, have been able to take that benefit of that, and they can walk on water if you wish. So they use that, they don't break the cohesion of the water surface, and they're able to use it for navigating over water sources. The next one that I want to mention is that water is excellent as a heat reservoir. So if you live by a big body of water, whether it's a big lake or by an ocean, you'll notice that the fluctuation in temperatures between the day and night are much lesser than it is in areas where we don't have these big bodies of water, such as here in the desert of Arizona. So in Arizona, we get really hot days and really cold nights during the winter. That's because we don't have that water that kind of absorbs the extra heat over the day and releases it over the night. So that's why often these coastal areas are very popular in terms of habitation, because the water helps to balance off those temperature fluctuations. The next thing that I wanted to mention about the water, we have been discussing traditionally about the three phases, three forms of the water. So how we have the solid ice, the gaseous water vapor, and then we have the liquid water that we know. There is a fourth one, but we're not going to go into that on this course. And the final thing that I wanted to highlight is that water is an excellent solvent. So a lot of things can dissolve into water, and this is why water makes such a good transport media, that we're able to dissolve things into it, and then those things get carried by the water. That works as our transport media to various places where we might need them. And there are certain kinds of solutes that do not fully dissolve into water but get transported still in the liquid, such as glucose, lipids, urea, creatine. These are not really huge interest to us. The ones that we are more interested are these that when they dissolve, they form as charge of some kind. So here, for example, we see many salts, acids, bases, and proteins forming a charge, and this charge will then have an effect on the behavior of these substances, so that allows us to predict what will happen. And there, I'm just simply highlighting that you see some examples of these charges. But let's jump forward. What's important is that some of these we find in different parts of the body. Some are within the blood, some are within this interstitial fluid, and some are inside the cells. They're not always in equal concentrations at the different places. So I'm going to show you a couple of important ones that we really want to know about. So here we see, for example, the sodium and potassium. So there's a lot of sodium both within the blood and in this interstitial fluid, but very little inside the cells. Whereas it's the potassium that we find in great quantities inside the cells, but not so much outside in the blood or in the interstitial fluid. That's because of these sodium-potassium pumps, for example, that we see this difference. The next one that I wanted to highlight here is going to be the chloride. We see great amounts of chloride, again, in the blood and interstitial fluid, but not really inside the cells. What I've noted here, we see the bicarbonate. So you remember we talked about the transportation of carbon dioxide in the blood as a bicarbonate. So there's some in the blood, also some in the interstitial fluid, lesser in the cell. And the final one that I wanted to show was these hydrogen derivatives, mainly within the cells. But really, that's as much as you need to know about those. The big take-home message of this discussion of the charges and so on is that whenever water moves from one place to another, then the electrolytes that are within that water, dissolved as a solvent, they move with the water, because water was such a good transport medium. So whenever we look at the movement of water, we have to remember that electrolytes will follow. Or if we look at the movement of electrolytes, we have to remember that it follows. So these are interdependent water and electrolytes. I have here a little bit of a diagram of some of the important sites where this exchange occurs, but we won't go into that on this course. What I do want to talk about is that we take in some water to our body. If you're wondering how much should you drink, you should drink about 2.5 liters in a day for an average sized individual. And we also lose some water. We lose about 2.5 liters of water in a day. So the water intake and output should be equal in a person throughout the day. Because if you take more in water than you lose, you would end up gaining water. And if you lose more water than you take in, you would end up losing water. And neither one of those is great in a long term. So we typically end up having an equal amount of water that we take in as we lose in a day. So let's have a look how we gain water. We mainly gain water through drinking. So the sense of thirst is the driving force for getting water into the body. Some water also comes from eating moist foods, and a little amount, maybe 10% of all the water in your body, as a byproduct of those metabolic processes. Like here we see the cellular respiration, the sugars with the oxygens being transported into energy and carbon dioxide, and as a byproduct, then we end up seeing these water molecules being produced. Well, the question that I have is then that how do we lose water? Well, we lose most of the water through urination, so the need of pee. Some water is lost in evaporation, sweating, and breathing, and a little amount, less than 6%, in feces. So the main driving force for water loss, water output, is the need to urinate. And for that, we do have a mechanism that's known as osmoreceptor ADH mechanism. I will mention that later on this class. You don't need to worry about it, and you don't need to know it in great detail at this point, as long as you know that such a thing exists. What I want to talk next is that in many textbooks, we get these complicated diagrams of how the water intake gets regulated.
Can you please go back?
I can go back, of course. Is this the right slide that you wanted to see?
Yeah, okay, now you can continue now.
Okay, yes. So I will continue, but I always say if I rush too fast. So in many textbooks, we see these diagrams that are really, really complicated. There's a lot of arrows, there's a lot of boxes, and you might end up thinking that, oh my goodness, this is too much to take in. So I don't want you to memorize any of these. You can see some diagrams that are less complicated, but still a little bit to do. They might look like seesaws sometimes. And sometimes you get diagrams where there's like two loops for two opposite events to happen. None of those I want you to memorize. We can do better than that, and we can do more simple than that. What we're going to do, we end up back to our good old friend, the concept of homeostasis, which is all about maintaining the consistent internal environment. And I highlighted at the beginning of our course that knowing the concept of homeostasis is so important because we will use it to explain many other things in A&P. So we remember this simple homeostatic loop mechanism diagram that we learned. There's a stimulus, it gets sensed, pass to the control center where we have the set point, and then some sort of an effector works to stop that stimulus from happening. Well, let's explain two situations where you have either too much water or too little water by using this, rather than using these complicated diagrams that I showed you from other textbooks. I'm all about keeping it simple. So the first situation that we're going to have a look of is what do we do if you have too much water in your body? So in this case, the stimulus would be that your blood is too watery. There's too much water in the body. That's the change that has happened, and your body always responds to changes. Well, how do we detect that change? We detect it with these structures known as osmoreceptors, which are located at the hypothalamus of your central nervous system, so the brain of yours. So there's these specialized structures known as osmoreceptors. They notice that there is too much water in the body. Well, the control center is, of course, going to be our brain. So brain then compares that to the set point, how much we should have water and how much we actually have water. Is it in balance? If it's not in balance, we need to do something. And what we do, we work through effector. And the effector in this case would be your pituitary gland secreting less ADH. So we're making less antidiuretic hormone. And you remember the function of the antidiuretic hormone was to play a role in absorbing back water in a nephron's distal convoluted tubule. Well, if we don't have ADH, more water will pass to the collecting duct and more urine will be made and we pee more, so we get rid of more water. So as a result of all this, we'll end up producing less urine and you get rid of the extra water through that. So that's what we do if we have too much water. And it's as simple as that, what I want you to know. And that's why the homeostatic loop mechanism comes handy for us to know. Let me know if I go too fast. Like I said, I don't want to keep you for too long, so that's why I move ahead. But if I move too fast on anything, just give me a shout. Now what we're going to do, we're going to look at the opposite situation where we actually have too little water in the body. And if we have too little water in the body, your blood becomes too rich in solutes. So you can say that the body becomes a little salty in that sense. So that's the stimulus, that's the change that's happening there. There's too little water, the blood is too concentrated, there's too much of all the other stuff than water in the blood. Well, again, we sense that with the same osmoreceptors from the hypothalamus of the central nervous system of the brain, and that passes the message to the control center, which is the brain at large. And there we compare it to what should be the normal amount of water in the body or normal waterness of the blood. And if it's not aligning with this set point, this normal, we act. And how we act is through, again, of this pituitary in your central nervous system. And now pituitary does make more ADH. And if we make more ADH, you remember ADH caused the distal convoluted tubule to become permeable. So more water is taken back to the body from the nephron, so less liquid is put out to the collecting duct, less urine is made, less pee is made. And if you make less pee, well, of course, then you lose less water. So this is the way how we can conserve the water, and then the water amount increases as a whole. So again, by using these simple diagrams, we're able to explain some pretty complicated concepts. So that was the big reason why I was so adamant that we are going to learn together the homeostatic control mechanism loop. And this is not the first time that we will see it. It's not the last time that we will see it. Well, now, if you're okay, I'm going to move ahead and talk a little bit about concept of tonicity. And tonicity should be something that you've seen before. So again, I'm going to go through this a little faster, but if you need more extensive explanation, you can give me a shout out, and I also provide that in the full lecture video. So we can have 3 different kinds of conditions, and we're going to go through those. And what we're doing here, we're looking at cells in a liquid that's either hypotonic, isotonic, or hypertonic. Before we start looking at that, we need to revise a couple of basic concepts. So whenever we have a lot of material somewhere, there's a high concentration of that material. It moves from the area of high concentration to area of low concentration through the passive transportation known as diffusion. So in this example, I have this drop of food coloring that's dropped into a glass of water. So the concentration of food coloring is going to be high here. But over time, it will just through the diffusion passively spread to all parts of this glass until it's even everywhere. So that's how diffusion works, and that's what naturally happens. We try to establish a balance so that there's no high and low area, but there's an equal amount of stuff on both sides. So in a hypotonic situation, we have a cell, and the inside of that cell contain a lot of electrolytes, or you can say that it's more salty. There's more salts inside the cell than outside the cell. Well, what your body will do, it will try to even out this difference. There's less salts here outside, more salts inside. How can we make it more similar? Let's add more water inside the cell. So water will be flowing into the cell to try to even out this difference. And as a result, the cell will grow bigger and bigger and bigger. And if it grows too much, it might even burst. It might break. So how do you remember that? Well, in A&P, in any biology class, I try to think of things that I find easy to remember. So when I hear a term hypotonic, I think of hypo sounds a little bit like hippos. And hippos are these big swollen animals. They look like swollen. So hypotonic cells are my hippo cells. They're big swollen cells. Water moves inside. Let's have a look at the next situation, isotonic cells. In isotonic cells, there's the same amount of salts both outside and inside the cell. So there's really no need to put in or take away salt. There is some amount, or take away water. There's some amount of movement back and forth, but whenever some water comes in, some water also leaves. So overall, the net change is 0. There's the same amount of fluid, the cell remains the same size. So how do I remember that in isotonic situations, cell does not grow and it does not get smaller? Well, I think of the term isotonic. That makes me think that I am so perfect, that I don't need to change. So isotonic cells are perfect cells. They don't need to change in terms of their size. Okay, let's look at the last case. In hypertonic cells, now we have a cell where the outside of the cell is more salty than the inside of the cell. Again, your body will try to even out this difference, and how it's going to try to even out is by putting water out from the cell to the outside. So water is flowing out from the cell, and as a result, the cell will shrink, and it might eventually even end up drying up. So how do I remember that hypertonic cells are these cells that shrink? Well, I think of what kind of people are hyper. Hyper people have a lot of energy. They run around a lot. And what happens if you run around a lot? Well, you become very skinny. So hypertonic cells are my skinny cells. They become skinny, smaller and smaller because they're so hyper. So here's just a reminder of those mnemonics that I use to separate between hypo, iso, and hypertonic. Hypotonic cells, my hippo cells, they are big swollen ones, so they swell. Isotonic cells, they're iso-perfect. They're perfect as they are. They don't need to gain, they don't need to lose, they remain the same size. And finally, our hypertonic cells, they were the super active, energetic hypers that became skinny in the process of being active, so they shrink, they get smaller. So that's how to remember the tonicity and 3 different types of tonicity. I hope that makes sense. So the next thing, if that makes sense, where I'm going to move on, we're going to start talking about what I said here earlier on about salts and having a look how that plays role in this. But before we jump into that, I'm going to ask you a question based on everything that you've learned here. Can you die out of drinking too much water? Well, what would happen if you drink too much water? Well, you would get hypotonic cells, they would swell, and they can eventually burst. And that's actually what has happened in some cases. There was a competition organized by a radio station, I think in back early 2000, I want to say maybe 2007. At that time, the Wii game console was new, and the radio station thought that it's going to be really cool to come up with a competition that we were going to name that Hold Your Pee for winning a Wii. And well, they did that. And the rules of the competition was that people phoned into the competition and they had to drink a lot of water in a short amount of time. They were not allowed to use a bathroom. So as a result of all of that, one of the persons who took part of the competition actually ended up dying because they should have gotten rid of that extra fluid. They did not. And if they did not, their cells eventually ended up bursting. So that's why everyone should take a basic biology class, even radio DJs. But anyways, so what I wanted to show with this picture was that sometimes when you exercise, or if you're out on a hot summer day in Arizona, you end up sweating a lot, and you might be drinking a lot of fluids, but you still don't feel totally well. You might get a craving for French fries, potato chips, something very salty. And that's your body trying to replenish these salts that we saw that could cause cells to become either hypotonic, isotonic, or hypertonic. So that's why the electrolytes, which are the proper version, we're not going to talk about salts anymore, we're going to talk about electrolytes. They are important. So what can happen, kind of summing up what I already referred to, you might exercise a lot, you end up sweating a lot, you drink a lot of water, and now the body fluids become too dilute, and to deal with that, your brain tells you to pee. And well, if you pee, you become more dehydrated. So even though you were just drinking water, you're becoming more and more dehydrated. That can happen. Well, how we avoid that? We avoid it by making sure that your drink contains electrolytes. You might have seen these electrolyte drinks somewhere else. There's many different kinds, Powerades, Gatorades, vitamin waters, and so on. So what we do with them, we try to replace the sodium and the chloride that we lose as we sweat. And there can be also water uptake enhancing elements such as the sodium chloride here. And I here collected a little bit of information how good different drinks are in terms of electrolytes. You'll notice that there's definitely differences. So these electrolyte drinks do a good job of replenishing those, whereas orange juice or soft drinks, not so much. Water, none at all. Well, that covers everything that I wanted to say about electrolytes and takes us to the very last bigger concept that I want to discuss on this chapter, and that's all about pH and pH scale. So just to revise, you should have seen this before. The pH scale runs from 0 to 14, 7 being neutral, pH value higher than 7, we call it alkaline or basic, pH value less than 7 is acidic. But what these numbers really tell us, they tell us the concentration of hydrogen ions in a mathematical formula or mathematical scale. So if there's the same amount of hydrogen and hydroxide ions, the substance has pH of 7. But if we have more hydrogen ions, now we have acidic substances. And if we have very little hydrogen ions, these become now basic or alkaline. So why does pH matter? Why do we need to worry about the pH in your body? Well, pH affects how your proteins function. Many biochemical reactions, and so on and so on. So we do regulate the body pH very, very closely, even normally. Normally, your pH of the blood is kept within the range of 7.35 to 7.45. That's a very narrow range. That's a very small variation in the pH, and your body does this automatically. Your body is pretty amazing, huh? Well, if your pH falls below 6.8 or gets higher than 8, you probably will die. So there's some room within variation of where we keep and before it becomes dangerous. But your body does this amazing job of keeping the pH so consistent normally. Well, if your pH is on the higher side, anywhere above the 7.45, we talk about a clinical case of alkalosis. And if it's below 7.35, we talk about acidosis. And you don't have to worry about alkalosis and acidosis. At this point, I will show what alkalosis and acidosis mean through signs and symptoms in a few moments' time. The big take-home message from this is just that remember your body does an amazing job of maintaining consistent pH value. And I see some of these weird diets, alkaline diets or alkaline ash diets or alkaline acid diets or acid ash diets or acid alkaline diets, all sorts of crazy diets. And some of these diets claim that by eating certain kinds of things, you can change your body pH. Well, now that you've taken this class, you know that that's nonsense. That's not true. Your body pH, if you're a normal, healthy person, will be kept within this very narrow range. How these diets work is that rather eating certain kind of foods that have a certain kind of pH is just in general better for you than eating foods that have a different pH. But you will not change your pH by eating food in a long term. It will not happen. So what I wanted to share with you now is that how are we able to keep this pH within such a narrow range? How do we do such a good job, and how can your body do this so efficiently? There's 3 ways how this can happen. First way is through various buffers. What buffers are? They are various different kinds of chemicals that bind with either acids or bases, and in this way neutralize them. These act very, very fast, so they just stop things from going acidic or basic by chemicals binding it. In my BIO201 class, we do actually test out different things, how good buffers they are. So that might be something that you remember from those classes. The second way, which is still kind of slow, but not super slow, takes about minutes, couple of minutes to act, is to normalize your body pH through respiration. So what happens here is that the center of the brainstem for breathing regulation detects that there is a hydrogen concentration either too high or too low. And in response to that, we then just change the rate and depth of our breathing. So we can breathe away hydrogen ions or we can keep them depending on what we want to do to maintain our body pH within this narrow range. Well, the last method that I'm going to show you is the most potent, most efficient of them all, but this can require anywhere between hours to several days to act. And that's, of course, the renal mechanism, so making pee and urinating out the hydrogen ions. And this is what we just studied in our previous chapter when we looked at the nephrons and urinary system. So this is how your body pH is maintained within such a consistent range. And final thing that I want to show in terms of the clinical relevance is what happens if you have either acidosis or alkalosis. You remember acidosis was having a low pH. So if you have low pH, your body starts to have certain kinds of symptoms. The ones that I want to highlight from here is that, for example, your nervous system becomes depressed. You might get confused, and in worst case, a coma might result. So, for example, in uncontrolled diabetes, an individual might go into a diabetic coma, and that would be when the acidosis happens. In alkalosis, instead, when we have high pH, your nervous system now works like hyper, overboard, overly excited. You get, for example, these weird tingling sensations. At worst case, you might get a paralysis. So that's why keeping the body pH within certain range is so important. And I promise this is the last slide of this presentation, this one. Well, this all brings us back. Why does this matter? Well, if you go to a hospital, one of the first things that we typically do is that we give you a drip. So we give you fluid therapy. This drip, this water that we give to you, saline water, contains just the right amount of electrolytes and salts. So basically, you could say that it's really expensive salt water. But what it does, it makes sure that you have right amount of these salts so we don't end up hypo or hypertonic. In a hypo or hypertonic situation, we instead remain isotonic. And that's really all you need to know about this chapter of water, electrolytes, and pH balance. And I know that I've kept you for quite a while, so I'm just going to jump back to the previous presentation. And this is not going to take a long time. We're just going to wrap up what we have seen. So let me jump there and share that with you. Here we go. You should be able to see the PowerPoint presentation any minute now. And we finished here looking at the key points of this week's chapter. We looked at the fluids, we looked at the electrolytes, and we looked at the pH scale. The last things that I have to share with you from this chapter or this session really is that do remember that if you need to retake assignments, you can message me with specific details. Usually the connect messages are the best. I know I have few messages there about reopening certain things, giving extensions. I will do that. I just wanted to get your exam and practical grade up first. I remind you that we have tutoring available this time because of the pandemic is still in effect. We do it all electronically. You don't need to have an appointment and you just use the Cranium Cafe service. Of course, I, as well as your instructor, I'm super happy to meet you. You just message me, we'll set a time together and we'll figure out whatever you want to figure out. We'll discuss it, either individually or as groups. And I've been doing more of those recently. I've been getting a lot of requests. And final thing, as well as always, if you have any questions, either you who are here with me today on this session, or if you're watching a recording, feel free to ask me now, or you can ask me through messages on Connect, and I'll definitely try to help you. But really, that concludes everything that I had scheduled for this session. And I just want to remind you, don't forget that you have the critical thinking question this week to do. It is not a hard assignment, but it's different from what you've done before. Just look at the grading rubric to figure out how many things you should find out and give it your best go. I'm usually fairly generous with grading that. But that's all I had planned for this session. Today, if you have any questions, I'm going to be here, happy to answer those. If not, I really thank you for joining me. I'm sorry that we ran a little longer because of reviewing the exam 2 and lab practical 2 questions. And that's
I have a question.
I'll try to help you.
Is it concerning the critical thinking?
Okay.
Yeah.
Do you
The first question? The
Oh,
first
you want to see
question.
about it? Let's jump in there. Let me
Yeah.
out of this. Ooh, I don't know.
I did the second one already, but the first one I'm trying to like grasp what it
Okay,
actually means.
let's
But
have a
I
look of
know.
that together then. Hold on a second. I will share with you my window and we can have a look of that together. Just bear with me. I'm jumping to the right material. Just a second, I'm pulling up the question and I will share with you my screen as soon as everything has loaded. Okay, now I should be able to share it. Let's have a look if you're able to see it. Where do I find
Oh yeah.
You should be able to see the critical thinking questions. That right? You're able to see your screen as well?
Yeah.
Was
Well, the
it
other
scenario,
question.
was it the scenario 1 or was it the scenario 2 that you had a question about?
The scenario 2.
Let's have a look.
The one was pretty explanatory.
Yeah, I think it was a little bit made more sense. So in scenario 2, what I'm asking you to do, there's a little bit of introduction there, and then I'm asking you to choose from these statements that is it likely to be true or unlikely to be true? Was there a particular statement or was it the next part of this that you...
Like the duralysis, like something like that. Can you explain?
Oh,
No,
is the individual...
no, not that one. No. Scroll up.
Scroll up.
Yeah, just to explain like the letters. I don't like
These
this
letters
one.
L and U.
No.
These
The
A, B,
number,
C,
like,
D.
yeah.
Go through them. Okay, so what
Yeah.
I'm noticing in this scenario, this woman entered to a competition to determine how much water could they drink in 1 hour. So this is the exact same situation that I told you that they had on that radio station competition.
Yeah.
After 50 minutes, what would we end up seeing? Well, the volume of...
Because you said the blood becomes watery.
It
They take too
becomes
much
watery.
water.
So basically, there's more water in the blood. So now the blood volume will increase. That's likely to happen. Also, the blood pressure will increase because there's now so much blood that we have nowhere to put it. So both of those will happen. The third thing, that she will have a diarrhea. She will want to, her body will try to get rid of that water in any way that it can. If she's hot,
I thought like diarrhea has to do with like passing out feces.
that would be, yes. But when your body is in a panic of trying to get rid of fluids, it's going to try to do it in any way. It doesn't have to be peeing. If your peeing doesn't work, then you basically pass water. That's what diarrhea...
Or like sweat out, right?
Yeah, so that would be likely as well. Things that are not true of these statements, that of course the water absorption will not be dependent on the glucose receptors on the intestinal epithelia, and there will not be any vasopressin, so those will not have...
What's the meaning of the vasopressin?
Vasopressin is one of the hormones that we might have touched upon earlier on. If we have not, basically what it does, it just controls, again, the blood pressure through our kidneys. But in this case, there will not be any, these critical thinking questions. They're kind of like pushing you to do a little bit of independent research, but really I can tell in this case, no, there will not be vasopressin. It will not be something that we release. What we will release instead is the natriuretic peptide. So you remember, we want to urinate now. Your body will start to make peptides, chemicals that make you urinate. And this is one of that's likely to happen. She will have a headache because you remember
I know that
went up so much. There's too much blood to fit into her cardiovascular system, and she will exhibit water diuresis. So there's too much water in her.
Like, what does diuresis mean? Like when it's too much water?
Yes, so for example, if she would go, for example, and have a pee, it's not that yellow urine, it's going to be just clear. So there's too much.
Clear, okay.
Yeah. And finally, the last one is that she will have more of the potassium in her urine than usual. That's not true. The amounts of electrolytes remain. She will just get rid of that water there will not be change in the electrolytes. Did that help at all? I kind of feel that I went through it quickly.
It helped because some words I was trying to like understand it, but
I
it helped.
totally get what you're saying. I think some of those words are a little bit challenging, and we're probably going to change this as activity for the next semester. But yeah, it's just a matter of taking a little bit of time, Googling them, looking them up, or like you did, asking from me. I'm totally happy to help. I want you to get full marks for this. So let me know if I can help with anything else. Was there anything else on this second question that you struggled with or found challenging?
We can scroll up. Do you want to scroll up?
Scroll up.
I just want to read through the question, the second question.
The second question.
Scroll down.
Scroll down. Yes, absolutely. I was like,
Yeah.
she wants me to go. So then I ask you to circle. Do you think that she's healthily hydrated or unhealthily hydrated? You could actually choose either one. I would be leaning towards saying that it's unhealthy, what she did. But someone can argue that that's healthy. But what I need to know your reason why you think it's healthy or unhealthy.
I think she's healthy because she has more of like, wait, let me see.
She's now drinking crazy amount of water in a very short amount of time, and we remember
Yeah, I
died
think she's
because
unhealthy.
yeah, it would not be healthy in my view. You have to come up with a great explanation if you tell me that it's healthy.
Yeah, that's
Some of the
true.
reasons why it's unhealthy is that the water will move now to the bloodstream, and as a result, your entire blood volume will go up, and your blood pressure will go up. Also, the fluid in her body will now become very dilute. So we remember that if it was very dilute, so now our cells end up swelling and swelling and swelling, and that's not good.
And they bust.
It's up to edema, and edema, one example of that would be headaches. And worst case, she can die. So I would be keen to say that it's unhealthy, but someone can argue that they are healthily hydrated. If they said that they're healthily hydrated, I would then probably think of that, okay, maybe they end up telling me that they will have a lot of high blood volume and high blood pressure and edema, but her kidneys will start working really fast. Her glomerular filtration rate goes up, and she will then excrete the extra water. But she just needs a little bit of time, and she will keep that sodium in her blood, and she will return to normal as soon as she's able to urinate it out. So it's up to you.
Yeah, if she can pass out the water faster, she should be okay.
She should be fine. The other option would be that she could take, for example, diuretics that make her pee, medications, or she could eat very salty food or drink electrolytes. And if you get that salty food or electrolytes, your cells will no longer swell. They will return back to the normal size. So it's just up to, that's what these critical thinking questions are, that you make up your mind and then you tell me why you think so. And you will do well if you tell me what you think.
Okay.
So there's no right or wrong, really. What I'm looking is that you just basically tell me what you think and you justify that.
Thank you.
Perfect. Is there anything else I can help you with? You feel quite happy?
Yeah, it's fine.
Okay, excellent. Well, I really appreciate you joining me, and I'm sorry we ran over our scheduled plan. And please do check your grades from the exam 2 and practical.
Yeah, I saw it.
Yeah, I think you're doing really well. Just keep doing what you're doing.
Thank you.
Okay, have a good.
Oh, can we
Yes.
see the labs because I can see what I go wrong.
Oh, let's have a look of that. Let me just pause sharing the screen. So we just resumed to wrap up this recording. So like I said, with the student, we just looked at some of the grade related matters, and I just want to keep those always between the student and myself, so we don't record those, we don't share those. But this really was all I had for this week's session. As always, if you have any questions, all you have to do is reach out to me and I will be more than happy to make sure that we get you your answers, we find you your answers. And I do want to thank you for being part of this class and for all of your hard work. I can definitely tell that this class is determined to do well and get good final grades. And I think we're well on our path there. So keep up your good work. Have a very good rest of the week. And remember that I'm here to help you. I want you to succeed. Until next time, bye.